I'd like to welcome our next speaker, Dr. Jose Luis Costa. Dr. Costa is currently the Director of Medical Affairs for the EMEA region in the Clinical Next Generation Sequencing Division at Thermo Fisher Scientific. Dr. Costa received his PhD in biology from Uppsala University in Sweden. In 2014, he was appointed an affiliated professor at the medical faculty in Porto, Portugal, where he retains his academic appointment today. Welcome, Jose.
Thank you, Amy, for the introduction. Hello, everyone. Thank you all for being here today. My name is Jose Luis Costa, and I'm currently serving as Medical Affairs Director for EMEA of the Clinical Sequencing Division of Thermo Fisher Scientific. It's really a pleasure to be here today and share with you verification data of our new assay for expanded carrier screening. As we all know, genetic disorders can be caused essentially by two large groups of variants: sequence variants and structural variants. However, the proportion of types of variants that explain a disease can vary. For example, beta thalassemia and cystic fibrosis are mainly caused by sequence variations, with copy number variants responsible for just a smaller portion of cases. In contrast, for Duchenne muscular dystrophy and alpha thalassemia, structural variants make up the majority of variants. However, if you don't look at sequence variants, you will miss approximately 25% of Duchenne muscular dystrophy variants. Sequence variants, either SNVs or indels, are typically detected by different technologies, including Sanger sequencing, genotyping solutions like microarrays, for example, and next-generation sequencing. On the other hand, structural variants involving large deletions and duplication events are typically detected by MLPA or qPCR. Traditionally, different technologies were required in order to detect the different types of variants. In order to avoid the need of different technologies, we have just recently developed an innovative new assay for expanded carrier screening based on the robust AmpliSeq technology. This one is called the Ion Torrent Carrier-seq ECS kit. This kit is a complete workflow that starts from genomic DNA extracted either from blood or saliva. Then it uses the AmpliSeq technology to generate the next generation sequencing libraries. Templating is performed on the Ion Chef, and sequencing is done using an Ion Torrent sequencer. Finally, analysis performed using a dedicated bioinformatics software for interpretation and reporting of variants. The solution maximized detection of couple at risk by analyzing full coding regions and then CNVs across all the genes that are targeted. This involved a rigorous design with specialized algorithms to address variants in challenging genes that are notoriously difficult to sequence. For example, the well-known SMN1, HBA1, and HBA2. This by consolidating standalone tests in a single solution. But why are these genes difficult? They are difficult because they share large regions of homology between them. And as we know, mapping short reads that come from NGS sequencer data is challenging when there is a high degree of sequence homology. This occurs between paralogs, such as SMN1 and SMN2, or between genes and pseudogenes, like as GATPH and CYP21A2, or within a locus itself, when it's randomly duplicated, such as in the region of HBA1. With carrier-seq analysis, there is a combination of sequence-based read copy number calling analysis and SNV genotyping, plus specialized algorithms to call variants in three classes of, let's call it, special case genes. One example is spinal muscular atrophy, targeting SMN1 using targeted copy number calling and genotyping to determine copy number of exon 7 in the SM1 gene. Second, similarly, is a special set of algorithms for calling CNVs in HB1 and HBA2 for alpha thalassemia. And finally, we have what is called the gene-con algorithms for gene conversion. In these genes with high homology, such as GBA, when looking at recombination events that creates a carrier status, when that gene has been converted in part or as a whole into the pseudogene, which is challenging to resolve without a special algorithm. An important novel aspect of this product is a newly developed algorithm for CNB calling. perform robust copy number calling across all of the 420 genes of the panel. There was an extensive optimization done on CNV baseline and also on the incorporation of relative inter-amplicon distance to determine the probability of an event occurring in the sample. R&D spent a lot of efforts working on the baseline, incorporating known source of variation. Multiple experiments were done manipulating the quantity of genomic DNA as an input, different PCR conditions, different library quantification methods, and then also incorporating the variation that is generated by unknown sources by, for example, doing multiple libraries, doing those libraries in multiple sites, and also by different operators. So all this effort improved copy number calling across genes, potentially even to the single exon level. And it also allows the detection of CNVs covering large genomic regions that span multiple genes, as in the case of HBA1 and HBA2. This slide is just to highlight the flexibility of the carrier-seq kit, the breadth of the solution, and particular aspects that are incorporated to address challenging targets. On the bottom, starting from the left, we have the panel that will be available supporting 96 reactions, and on the different sequencing capacities using the different SIP solutions that may be available. In terms now on the panel content, it is a two-pool AmpliSeq panel, and we've gone through extensive design iterations to improve this panel, in particular, again, the copy number calling. This is just a list for those that have their own special disorder or gene. But be aware that this is just a partial list of some of the more relevant targets for expanded carrier screening. Note that in red font are the special case genes that I've just discussed briefly before with dedicated algorithms, and they are noted with the abbreviation SC as for special case. We make special claims around those genes and what we can detect because of the challenging nature of the genes, for example, CYP21A2, TBPA1, HBA1 and 2, and SMN. Also on the list, I marked the genes with yes plus in green, which are the ones that had extra design efforts to enable exon level copy number calling. Extra amplicons were added to provide more robust copy number calling down to the single exon level for genes relevant as CFTR and DMD. As I said before, the total content is 420 genes, but we actually included 25 base pair flanking regions intron-exon boundaries to detect splice site variants and also intronic variants. That breadth of coverage and the number of genes resulted in a very large panel of 400,000 amplicons. Also importantly, incorporated in this panel are sample ID amplicons for gender determination and provide sample tracking to detect possible sample mix-up that might occur during the laboratory process. Overall, we target over 36,000 CNVs and indels from ClinVar and private variant sources. We've also spent time designing more amplicons on certain genes that are 33 of them, which are calling focused CNV targets. Importantly, this panel, as I said, has gone intensive testing using more than 450 positive samples and 68 synthetic variants. And this is what I will show next. As I said in the beginning, extensive verification and optimization was performed for this panel. This table sums up the verification and validation data showing performance of the panel. Performance was obtained over 242 libraries that resulted in 96% of the libraries passing our QC and accepting requirements. Additionally, both intra and inter-run libraries were above 99%, meeting the specification, and uniformity was greater than 95% of the bases. As you can see, CNB calling of carrier positive and controlled samples were higher than 99%, which is similar for CNB calling, with particular concordance for special case gene variants detection like HBA1 and 2, GBA SM1, and SMA silent carrier status. Here now we show the data using a chorial cell line known to be an SMA carrier sample with a deletion of one copy of SMN1 gene. Looking at the plot, we see that most amplicons support an average copy number of 1.5, whereas distinguishing amplicons, the ones in green boxes, resolve this as 2 copies of SMN2, the blue triangles, and 1 copy of SMN1, the red circles. This happens because ion reporter software and the carrier report software interpret amplicon representation to call copy number variations in the genes and annotate the significance of variants respectively. Normalized read counts for each amplicon are compared to the normal baseline, and an algorithm is then used to call copy number and functional copy number for both genes. Most amplicons and their primers are completely identical between both, and thus represent the average copy number of both genes. However, several amplicons in and between exon 7 and 8, the ones in green boxes, distinguish between the 2 genes and are exclusively amplified for SMN1, thus the result. When we look now at the more extended data, here is the analytic evaluation of 91 genomic DNA samples for the detection of SMN1 and 2 variants and copy number status. This evaluation resulted in high levels of sensitivity and specificity, but most importantly, the copy number accuracy was higher than 95% for changes in both genes, demonstrating the robustness of the ion-tolerant carrier-seq kit. This slide is just for those who are not familiar with the workflow to help you understand the length and the various steps of both library preparation, templating, sequencing, and analysis across the various S5 instruments. On average, the entire process for 32 samples takes around 3 hours, but importantly, only 36 hours. But importantly, only 2.5 of those are really hands-on. Of course, the analysis time will depend on the instrument. Some instruments will analyze the results faster than the others. The data analysis pipeline starts within the ion-torrent suite software for run planning and monitoring. Then the ion reporter performs alignment and variant calling for SNVs, indels, CNVs, and gene conversion events with the pre-configured workflows that incorporate all the special algorithms that I've discussed previously. And then the data is pushed into the carrier reporter software for automated variant classification and carrier analysis for reporting. It is important to note that carrier-seq uses the latest human genome build. Also, the dedicated ion reported workflow incorporates all the optimized algorithms. And we have established and incorporated acceptance criteria highlighted in the table. These are mean read depth with an acceptable value of more than 200 times coverage, and uniformity threshold that should be greater than 93%. And finally, reads must have a minimum length of 155 base pairs with an AQ20 quality score. All this information can be found in the ion torrent suite run report. A reporting solution is included and is fully customizable. You can customize the genes to include in the report. For example, you may want just to report on a subset of the 420 genes that you consider most relevant. You can also customize the variants to be reported. For example, include or not the nodule mutations. For important variants, you can select the variant by clicking the star icon and turning it into a color purple, as you see in the example. And this would generate an alert for the need for confirmation test if this particular variant is not properly sequenced. A good example would be the Delta 508 variant in the CFTR gene. And you may customize your reporting by not reporting, for example, benign or likely benign or variants of unknown significance. Also to note, if you are familiar with spinal muscular atrophy and SM1 screening, there is a condition called 2+0 silent carriers. These carriers have 2 copies of SM1 on one chromosome and a deletion on the other. The copy number is 2, so it looks normal, but they are carriers. And we have introduced a mitigation strategy because it has been reported there are SNPs in linkage disequilibrium with a 2-copy chromosome. This also can be configured in the software to be ethnicity specific and flagged when occurring. In closing, I want to highlight the solution outcomes and benefits. One of the key features is the full coding region coverage for the 420 genes, for CNVs and indels detection, but most importantly for CNV detection, improving the capacity to detect couples at risk for passing down serious conditions. The benefit for the couple is that they have the ability to pursue alternative actions, such as prenatal diagnosis or IVF, if they have an identified to be at risk carrier couple. Another valuable feature is incorporating the difficult to detect targets to homology in a single assay. This consolidates multiple standalone tests for these technical challenging genes with the benefit of improving the efficiency of the laboratories, both in terms of cost, but also in terms of time to results. And the effort that we've been spent in creating the features of optimizing the kits and the software together for a single solution provides an outcome of a streamlined implementation that helps ensure reliable and consistent results. This allows the implementation of carrier-seq in less specialized labs and enables carrier-seq testing across the globe. And with this, I thank you all, and I'm here to answer any questions that you may have. Thank you.
Thanks, Jose. Very, very nice presentation. I have a couple questions for you. How are the 420 genes selected?
Yeah, thank you. That's a pretty good question. So the 420 genes were selected essentially on the penetrance of the disease, on the severity of those disease, and then of course reaching out to the scientific community to get their feedback on who would be the most important genes to be incorporated into the design of the panel.
Yeah, that's key. With the screening, the carrier screening software itself, is it possible to configure what variants are reported on, or do you need to report all the results from the assay?
No, the entire software is fully customizable, both from the point of what genes you want to report in. You can just select your favorite gene to report, but also you can also select which variants from that gene you want to report.
Oh, that's nice.
Both on the particular variant itself or on the consequence of the group of variants. For example, you can specify a specific mutation that you want to report or not, or just as a group, like do not report benign variants, for example.
Similarly, I just one follow-up while we still have time. On variants of unknown significance, how are they being treated in this platform? Can the platform select to report variants of unknown significance or not?
Yeah, that falls into the big or in the different classes of variants that one can report. Benign, pathogenic, likely pathogenic, and then there is one class which is the variant of unknown significance, which normally it's quite a large class. And again, it's fully customizable so that you can select to report it or not.
Great. Thank you so much, Jose.
Thank you. It was a pleasure.